For this class, we're discussing a merge between biology and chemistry, which is actually my favorite mixture. For now, we're going to study the types of proteins, which are the globular protein and the spherical protein. For sure, there is more that awaits, but for this class, we're discussing these types. For the globular protein, from its name, you can obviously know that it's globular, it has a ball shape. And this is kind of its description. As you can see here, it is coiled in a spherical shape. Like it's not helical, it's not spiral, it is spherical. And it's also soluble. By soluble, I mean that it's a polar molecule. Like it can form hydrogen bonds with water and dissolve inside it. It has its hydrophilic R groups. We know, of course, that some amino acids have R groups that are hydrophobic and others have hydrophilic. For the R groups, we know that they have the alpha carbon attached to a hydrogen and the R group, that's a variable group, with the carboxyl group and, of course, the amine group. This is the structure of the amino acids. Okay, and this R group is variable. But what I'm saying is some can dissolve in water and others not. Okay, so the globular protein does a very special thing here. It hides, like they're hidden inside. The hydrophobic ones, which are enemies of the water, they're hidden inside. While the hydrophilic R groups of the amino acids are facing outside. They're facing the water, which is friendly with them. They can react and interact together with nothing of the problems going on. Okay, so by soluble, we said that its hydrophilic R groups are facing outside, cytosol and aqueous environments. And the hydrophobic R groups, like they don't like the water, are facing inside to be able to form the hydrogen bonds with water. They have a tertiary structure having a three-dimensional specific shape, and this makes them metabolically active. By metabolically active, I mean they can interact and interfere in chemical reactions. Like the globular protein has some examples as enzymes. Enzymes are not there for their shape. Of course, they have an active site that determines its shape, so its function, but it's actually mainly a functioning metabolically active structure. Okay, the enzymes, they break down and catalyze their reactions, okay, in the body. So these globular proteins are metabolically active inside the body. They have a tertiary structure, which we discussed the 3D shape, yet some of them have the quaternary structure, which is of many polypeptides that are together, not only one, there are many. Okay, that's for the globular protein part. About the fibrous protein, as you can see in the diagram here, it's not as spherical or globular as the globular protein looks like. It's long and its polypeptides are parallel to each other, which means they cannot meet. They're parallel, okay? Of the polypeptide chain having a large number of amino acids. And also, which we forgot to mention, that it has a regular or a sequence of repeated amino acids. Like these are not randomly. If I can find a chain of, for instance, alanine, cytosine, and glycine, for instance, at every third amino acid, I'm going to find glycine. One more time. Okay, so glycine is going to be found at every third amino acid. And that's what we're going to discuss in the collagen structure. Okay, so it has a sequence of repeated amino acids. It's not randomly placed as the globular proteins. It's sequenced. Okay? Also, it's quite the opposite of the globular protein. It's insoluble, where the hydrophobic, which do not like the water, are laying outside. They're facing the water, which of course cannot interfere or interact together, so that keeps it precipitated and insoluble in water, and of course in blood too, so it cannot be transported well in it. Okay? And this one in particular has a structural function. As collagen, we care for its structure, not for its function, or like it doesn't perform something as important as the globular proteins, okay? Also, it's more resistant to changes in pH and temperature, which means it's less sensitive to them. Like it can be facing like such pulling force and pressure, pH and temperature, and it can be more resistant. But if you have a 3D shaped ball and you squash it, it's gonna be everywhere. You're never gonna see it. But if it's a 2D shape, it's gonna still continue to exist, okay? And this one also has no tertiary structure. As I said, you can imagine it, or you can say that it's a 2D shape, maintained by bonding between R groups. Like the bonding between the R groups is not tertiary, okay? That was for the fibrous protein, and as you can see, it has some parallel polypeptide chain. For some examples to apply on what we've studied, we have collagen, which of course is very famous for anybody interested in biology. Collagen is an example of fibrous protein, which we've just said, it just has the same properties. It's insoluble. It cannot form hydrogen bonds with water. It's long with a repeated sequence of amino acids. Okay? But it's actually formed on 4 stages. The collagen formation is divided on 4 stages. It comes as polypeptide helix shape. Then we have the collagen molecule itself, then the fibrils, then the fibers. Okay? Now let's read together and try to understand. We have the polypeptide helix, which has a helix or a spiral shape. Okay? As you can see, we're here in the diagrams. It's made of glycine amino acid, and of course, much more types of amino acids, but its main glycine amino acid is found at every 3rd position. Like if it's a chain like that of different amino acids, like you're gonna find the same type at every 3rd position. Keep counting and you're gonna find them. Okay? Then these polypeptide helix, we have a triple helix polypeptide that forms the collagen molecules. We have 3 polypeptides that are held together by hydrogen bonds, forming a triple helix. Then we get these collagen molecules and we attach them together. They're parallel and they're cross-linked. They have the cross-links, and this is exactly what I mean. They are all parallel to each other, and they have also what we call staggered ends. These staggered ends, which means like it doesn't have an end. There's something hollow here, and these spaces, it's not like regular or something. Okay, so that's what we call staggered ends, and that's to avoid having weak points between the protein structure here of collagen. So these parallel ones that are cross-linked together by covalent bond, and this forms what we call fibrils. So this is a collagen fibril. And after that, we have the collagen fiber, which is what we want, the collagen fiber. And this collagen fiber have a high tensile strength that can withstand the pulling force and the high pressure. So let's recap again. We have a polypeptide helix, of which it has the glycine amino acid. It's also a long chain. It has the glycine amino acid at every 3rd position. Okay? Then we have the collagen molecule formed of a triple helix. Of them, they are attached together by hydrogen bonds of 3 polypeptides, triple helix by hydrogen bonds. Then there are many covalent bonds of attaching that many collagen molecules attached by cross links while they are parallel to each other. having these cross-links and attached by a covalent bond. And this is what we call fibrils. Okay? Many fibrils lie side by side and giving this tensile strength, which makes the collagen as a structural functional protein a strong one of its type. It can withstand the high pressure and the pulling force on it. And now that was for today's class. Thank you guys for watching.